So, yeah, I think we can probably start by now. Yeah, so it's my great pleasure to welcome Jami Saloman with us today. Jami volunteered to be, I think, just the second or the third external speaker in this seminar series, and Jami is one of the I would say most prominent leading figures in the international field of cancer neuroscience, and you can see this from all the articles she's publishing, reviews, opinion papers, perspective papers, original work, 2016. She had an extremely important PNAS paper where she describes the influence of sensory neurons on pancreatic cancer development and growth. And yeah, so Jami is leading a lab or labs at the University of Pittsburgh, and was closely working together with her, I think, old mentor, Brian Davis, who's also at the University of Pittsburgh, a neurobiologist who got interest in this direction. And her lab focuses on nerves and cancer. And Jami is not a CNS cancer person, but a PNS cancer person, so to say, and not brain tumor, but pancreatic cancer mainly, and other cancer entities. We just discussed that shortly last 10 minutes, that now, of course, is the time to develop methods for cancer neuroscience to probe neuron cancer interactions throughout the entire body. Yeah, because it's clear the body is full of nerves. The cancer can grow everywhere in the body. So now the big question is, how are these concepts that we discussed last times are reflected in, well, we as brain centrist people say peripheral, but you can also say most important, because these cancers are much more frequent than what we are talking about in brain tumor research, right? So yeah, as I said, Jami is a leading figure here in the field, and it's really great to have you with us. Jami is also a member of the Pittsburgh Center for Pain Research. So she's not doing just cancer, but also pain research independent of cancer. And also, and this is probably most interesting for us, is a member of the Biobehavioral Cancer Control Program. Biobehavioral Cancer Control Program. So that means really a system biology approach, something that is certainly needed for cancer neuroscience at the Hillman Cancer Center in Pittsburgh. And yes, Jami, I've talked enough. We are looking forward to your talk now. Thanks for coming.
Thank you. That was a very gracious introduction. A little flustered. Anyway, I wasn't sure what everyone's background was, and I know there's a lot of people there that are CNS tumor people. So I sort of broke my talk into a couple of different parts with some more overview type information just at the beginning to sort of set the stage. So I'll just quickly review the PNS for anyone who that might not be their area of expertise, and some of the key findings that have developed over history about tumor and nerve cell interactions. And then in the second half, I'll focus more on my own work and the sensory neurons regulation and the role in pancreas cancer. And just feel free to ask any questions or stop me, you know, either unmute yourself or use the chat. I'm happy to do that throughout the talk. I do just have one disclosure. I'm not going to show any data, but I do have funding from Signal Therapeutics for doing RNA-seq on tumor innervating neurons. So that is in progress, and hopefully we'll be able to share some of that in the coming months. But to get started, this is a simplified diagram of the peripheral nervous system. And so as you can see, it's not simple at all. And there's many different departments, compartments, and types of nerves that innervate all of the different organs. And we like to think of two main subdivisions of the PNS. and the first being the somatosensory system. So that's the one that I focused on historically, where your different organs have sensory neurons that are carrying information into the CNS. And they're not only carrying information supraspinally, but then also sending information back out through motor neurons to those organs. So that's pretty straightforward. These cell bodies of these sensory neurons are located in dorsal root ganglia that are within this vertebral column just outside of the spinal cord. The other compartment or division of the PNS is the autonomic nervous system. And that can be a little more complicated because it has multiple subdivisions. You may be familiar with parasympathetic. And those are the motor neurons whose cell bodies are in the brainstem, and they project to the different organs, mostly thoracic and visceral, very little somatic innervation. And they travel in this vagus nerve that has many branches going off to all of the different visceral organs. And they synapse either in organs on postsynaptic neurons that are in little ganglia within or near the organs. But one thing that I think is really important to remember and that makes the PNS complex is that this vagus nerve is carrying those motor axons, but it's also carrying axons from sensory neurons whose cell bodies are in the nodose ganglia. So even though the cell bodies are located in separate places, they're all traveling in the same nerve. The other major compartment of the autonomic nervous system is the sympathetic nervous system. And these cell bodies are located in the spinal cord. And when you zoom in, some of them are coming out, the axons are coming out, and they're synapsing in that sympathetic chain of ganglia that's located just outside the vertebral column. And there's also some that are synapsing on neurons and ganglia that are located near the visceral organs. So for example, we'll talk about later the pancreas, there's sympathetic neurons that are located in the celiac ganglia close to the pancreas itself. But as you can see in the mirror side here of this blow up of the spinal cord, that somatosensory system that we talked about is actually traveling in the same physical structure. So even though we think about them as two totally separate parts of the peripheral nervous system, they're really mixed together. So that's the brief introduction, just review of the peripheral nervous system. And I think the first thing that we as peripheral tumor and cancer neuroscientists think about is the fact that these tumors that are non-neural tissue get innervated, and just like the normal healthy organ, axons go to the tumor itself. And there's many different ways to quantify that. So typically, clinically, you get a tissue sample or a thin tissue section from a patient, and you stain for some non-specific marker of nerves. And some scientists will then count the individual axons that they see throughout the stroma and around the tumor cells. Others will measure the larger nerve bundles or the fascicles that contain multiple axons, and they'll either count them or measure the cross-sectional area. And in head and neck cancer, it's very common, and also in animal models, when you have a tumor that you have implanted, they'll measure the distance from the edge or the leading edge of the tumor spreading to the nerves. Now, of course, you might get slightly different answers depending on which quantification you choose, but in general, we've shown in many different types of cancers that when you have a normal healthy organ, and this is a cleared healthy tongue that's stained for nerve fibers, there is dense innervation. But when you then have a tumor present, that innervation becomes significantly greater. And you can see that there's a lot of sprouting of these nerve fibers. And clinically, this increased innervation is correlated in most cases with worse prognosis. So there is an active process of the nerves going towards the tumor. A whole separate group of scientists in this area are now studying the reverse interaction, where they're looking at the tumor cells going towards to the nerves. And that is called perineural invasion. And this was first actually reported in 1835 by a French pathologist who described a mammary tumor. This is a cartoon of his description that was then growing along the facial nerve. And actually you see individual tumor cells penetrate into the different layers of the nerve and actually get inside there and then start growing. And this also, like the tumor innervation, the more perineural invasion you have, the stronger negative predictor it is of the cancer prognosis. And this perineural invasion is actually most common in carcinomas. You see almost 100% incidence in pancreatic cancers and head and neck cancers, and that's where a lot of the research has happened. But it is commonly seen in many different types of other peripheral tumors. And so yes, we see these two nerves like going towards each other, but for many years it was thought that this penetration into the nerve was really some action by the tumor cell and that the nerve was just passively being attacked. But over the last recent decades, there have been several studies out of multiple labs that have shown that this perineural invasion is an active process and that the nerves are actually engaged. So in the periphery, the actual axonal fibers are surrounded by myelinating Schwann cells. And this is a beautiful video out of Richard Wong's lab showing in green that these Schwann cells are actually reaching out from the nerve and grabbing the tumor cells and pulling them into the nerve. So this is just a beautiful illustration of the fact that it is this active dynamic process. And there has been several reviews that have gone over the different cellular mechanisms that are implicated in this process. And I guess not surprisingly, it's been many different sorts of chemokines and peptides that are involved in chemotaxis and guidance, growth, adhesion. But what the point is that these studies have shown that it's an active engagement between the neurons, the other cells in the area, as well as the Schwann cells and the tumor cells itself. So this is really a partnership happening between these. And of course, not only is it going to cause sensory or sensory motor deficits and neuropathies, but this is a way for the tumor to leave the primary site and metastasize. So if we can intervene in some way in this process, it can improve responses to cancer therapies. So how I got involved in understanding this and being my interest in this was these are sort of innervation and perineural invasion are happening once there's a tumor there, but are the nerves important in even the initial development of the tumor? And when you look back over history, going back well over 100 years, there are findings that suggest this is the case. So going back to 1903, Dr. Cheadle described several case reports where he looked at the cancer incidence or the site of the primary tumor, and it was always in an area that was highly innervated and there was lots of trophic factors present, suggesting that there's some connection there between the nerves and promoting tumor growth. And so interestingly, in the 1950s, I'm not sure how many people are aware of these studies, they're not really ever talked about, but Dr. Scherer, who studied cockroaches, actually went in and transected the nerve and showed that it caused the induction of gastrointestinal tumors. So here is an incidence, it's sort of going against Dr. Cheadle's hypothesis that the trophic factors from these nerve areas are important. But it is suggesting that the nerves have some active role in tumor growth. And this studies like this continued for decades. And the first time that someone, instead of going in and surgically cut a nerve, they used a chemical to kill certain neuron types was 1979, where they did a chemical sympathectomy. So they injected a drug called 6-hydroxydopamine, which kills neurons expressing the dopamine transporter. And this, in their model, actually slowed the prostate tumor growth. So we've been going back for decades with these studies that have really implicated nerves in cancer, but it didn't really come to the forefront or emerge as a key field of study until more recently, with 3 very high profile papers that came out in the 2010s that all demonstrated neural regulation of non-CNS tumors. And all of these papers are very detailed with many, many experiments. So I'll just go over the quick highlights to demonstrate what the key finding was. So this is one technique where if you use tumor cells bound to luciferin, you can actually use a luminometer and in vivo track the tumor growth and metastases. And so coming out of the late Paul Furnet's lab, they used the 6-hydroxydopamine method to sympathectomize the prostate, and were able to show that this prevented the tumor growth and metastases in this model. And they were able to replicate this finding if you cut the hypogastric nerve. I think personally that the most important finding from this study that we really need to investigate further is the time course. So they found that if you do this deinnervation early in the animal process, either neonates or young adults, you are able to significantly prevent precancerous neoplasias and tumors. But if you do this later in life, it doesn't really have any effect. And this is probably the first time that there's evidence to suggest that the role of nerves in regulating cancer has time dependence, and the point at which you manipulate them is important. And this is just demonstrating, confirming that if you delete receptors for the primary sympathetic neurotransmitter, norepinephrine, you have the same effect and can reduce the tumor growth compared to a wild type. This is not unique to the sympathetic nerve. There is an importance of other nerves as well. And the following year, coming out of Tim Wong's lab, they focused on the vagus nerve and the parasympathetic motor axons, and showed that if you denervate the anterior side of the stomach, you don't get tumors, whereas the posterior side that remains innervated gets tumors. And this is just quantification of that. And demonstrated that if you delete the receptor M3, which is a receptor for the primary parasympathetic neurotransmitter acetylcholine, you have that same reduction in tumor growth. The key finding that actually came from his study is not just that transecting the vagus nerve regulates cancer, but that it's in a tumor-specific manner. So in a later study, they repeated that same surgical process of cutting the vagus nerve, but in a model of pancreas, and actually showed the completely opposite result, that a vagotomy increased the incidence of precancerous lesions and of tumor incidence. And so, as Frank mentioned, my focus is really on sensory neurons. So I would be remiss if I did not point out the seminal paper implicating sensory nerve regulation in cancer. And this was looking at, here on the left, basal cell carcinoma model. And what you can see in this graphic is a complex large basal cell carcinoma. And on the right, in a denervated animal, it's much smaller and less complex. And this is the quantification on the bottom, suggesting, again, this early sensory denervation, in this case of sensory nerves, is important for tumor growth. But just a couple of years later, in another model of skin cancer, this time melanoma, coming out of the Bunamovich lab, they decided to do their nerve transection right before they induced the tumor. So either the day before they induced the tumor or even a few days after. And you can ignore all the lines in the middle. The main point is that in control animals, in this black line, you have a normal tumor development over 2 weeks. But in the transected animals, whether you do it right before or right after the induction of tumor, you exponentially increase or accelerate tumor growth. So this is another piece of evidence that not only are sensory nerves important also, but that either there's a tumor or time-dependent effect on what they're actually doing to the tumor growth. So based on these studies, it was important to me, the takeaways that I came up with is one thing that we don't talk about enough is that all of these nerves are mixed nerves carrying both sensory axons and sympathetic, or sensory axons and like parasympathetic. And so when we do some manipulation to target the neurons or the axons that we care about, how is that affecting the other things? And that's still a huge hole, even in my own research, that I think we need to address in the future. But mainly these studies show that there's dynamic interactions between the tumor cells and the neural cells, and that the peripheral nervous system is contributing at different time points all throughout the disease progression. And so these are the studies and the history that inspired my own research and how I got to be a part of this field. And I came to my postdoctoral training to study pancreas cancer pain, and ended up studying pancreas cancer. And so for anyone who's not familiar with the pancreas or its function, it's a gland in the viscera that is made up of two types of secretory tissues. Endocrine tissues, which are like islets, releasing hormones important for glucose metabolism. And then the majority of the tissue is actually made up of exocrine tissue, where you have these acinar cells that release your digestive enzymes, and they're carried through the ducts to your gastrointestinal tract to help break down your food. And just like any other organ in the body, you have blood vessels and lymphatic innervation of the tissue. And that's how originally most people thought the tumor cells would get out and metastasize. But we now know that there is perineural invasion that also occurs and could contribute. So just to simplify the nervous system or reiterate the innervation of the pancreas, you have the two main sources of innervation, the vagus nerve that's carrying both the parasympathetic and sensory neurons. And you also have spinal innervation where you're carrying sympathetic information as well as sensory information. And this primary nerves carrying that is the splanchnic nerves. So when you stain a human pancreas for just general nerves, you can see here that there's a very dense innervation and that it's going to both the exocrine tissue and synapsing on the islets. And within the pancreas itself, there's also these small groups of neurons called interpancreatic ganglia, which are also receiving input from that external innervation. So, what does that mean in terms of this neurocommunication within the pancreas? So it means that you have all different types of fibers, and there actually is anatomical and functional evidence suggesting that they innervate all the different types of cells within the pancreas. And when you look at what they express, I think it's important down here to recognize from this nice review that many of these different fiber types that have different functions utilize the same signaling pathways. So that's important when you're trying to do pharmacological interventions or manipulations, because that also, just like they release many of the same signaling molecules, they express many of the same receptors. So that's the brief introduction of the pancreas, and now moving into the cancer part of it. The pancreas cancer incidence is 1 in 10,000 people. But 95% of those tumors are what we call pancreatic ductal adenocarcinoma, meaning that they're deriving from those exocrine, acinar, and duct cells. And there's a very low survival rate. And the reason for that is because it's rarely detected early. So it's not typically detected until patients are late in disease and they come into the clinic with pain, jaundice, significant weight loss from malabsorption and dysfunction of the pancreas. And like many other cancers, there is an inflammatory response that accompanies this tumor growth. And what's thought to be happening is that there's your normal epithelial or acinar and duct cells start differentiating. And you have these low-grade neoplasias that are really benign and actually develop in everyone as we age. But in some subset of patients, they go on to become malignant, and you have high-grade neoplasia. And then when they break out of their little lobe, because the pancreas is lobular, it becomes this invasive carcinoma. And so what do we know about the neurobiology of PDAC? Well, sure, many of you are familiar. There are some of my own personal mentors coming out of the groups in Germany that have done beautiful characterizations of specimens from patients looking at the neural changes that are associated with PDAC. And they have characterized structural changes where you see sprouting of axons and increased innervation, just like we've seen in the other tumors that I talked about. You also see neuritis, and so that's associated with some destruction and damage to the nerves, as well as invasion. So you see both immune cells and tumor cells getting into the nerves. pain, as we already talked about. And then they've done many, many studies demonstrating that the signaling molecule is important for neurotransmission in the periphery, are dysregulated and often upregulated in these tumors, including the neurotrophins, ion channels that are associated with hyperexcitability, cytokines and chemokines that are associated with the immune response. And so, this is clear evidence that nerves are important in PDAC, but what we can't get from the patient data is what's happening early in disease and how is that developing. And so that's why we have to move to animal models. And that's where my lab really focuses. So the animal model that we use is called the KPC model. And normally in a wild type mouse, the pancreas hangs off the stomach and just sort of floats in the mesentery there. And in our model, we use a exocrine cell-specific Cre promoter system to drive a gain of function mutation in KRAS and a deletion of the P53 allele. And we introduced a fluorescent reporter gene. So now when you open the mouse, the pancreas is nice and pink, and under fluorescence, it glows red. And so just like the cartoon diagram that I showed you earlier, in these animals at about a month, you start to see these low-grade pannins with a little bit of inflammatory stroma that becomes much more pronounced as they move to malignant neoplasia, these high-grade pannin lesions, and then to invasive carcinomas. And this takes median survival time is usually 5 months, where you start to see morbidity. And at that 5-month mark, about 30% of the mice actually show gross metastases. And what's great about the fluorescent reporter is it allows us to really visualize these metastases. So you can see little mets all along the spleen here in this particular example. So we started out characterizing this model by looking at the tumor time point. And just like what we see in the other models and what we saw in the human cases, we see hypertrophy of the nerves and sprouting. We see significantly greater innervation of the tumor than we do of a control pancreas or healthy pancreas, which I'll show you in a second. And so as you can see here in the merge, that the ductular tumor structures are very closely integrated with the nerves. So just like in patients, we are seeing the nerves go to the tumors, but what about the tumors going into the nerves? So we do also see perineural invasion. And if, for example, you look in this case, the tumor encases the dorsal root ganglia that's carrying the sensory fibers and the ventral root carrying the motor fibers. And when you look into the ganglia itself, you can see the individual tumor cells. And here where we've counterstained for the neurons, they're literally squeezing between the neurons to get in there. And this is not restricted to the sensory system, of course. If you look at the sympathetic celiac ganglia that is near the pancreas and carrying those sympathetic cell bodies, you can see that there's a loss of the neurons and replacement by tumor structure, and there's still close apposition to the neurons that are left there. But when you look at those neurons, they're not normal, healthy neurons that are left. They're expressing markers of nerve injury, such as ATF3, suggesting that the tumor is causing significant injury and other neuropathy. So that's important. We're showing at the tumor stage what we see in the patients, what's seen in other types of cancers, but we're still not sure at this stage what's happening early in disease, and is the neurons in any way contributing to the disease development. And so we started looking at earlier time points. And this is an example where you can see the nerves are present in a control healthy normal pancreas, but they're very small, sparsely labeling here and thin. And when you start to move into this 6 to 12 week time point where there's those neoplasias, but we still don't have a primary tumor, you can see that there's increased growth and the size of the nerves are getting bigger. And when you move into the neural tissue itself, again, there's no tumor at this point. When you look histologically at the pancreas, you do see individual little CT tomato cells that are penetrating into the DRG, and in this mouse, even into the CNS and migrating into the spinal cord. So it is clear that there's interaction potentially is preceding the formation of the primary tumor and potentially contributing to it. So as we talked about earlier in the talk, the idea that the primary tumor is developing because of the innervation and high neurotrophic area, we've looked at the expression of the neurotrophin signaling axis in these mice. And you can see that with age, as the disease progresses, the expression of both the nerve growth factors as well as their cognate receptors significantly increases. But this doesn't, we've taken the whole tumor out of the mouse here. So there's nerve terminals, there's stromal cells, there's immune cells, there's tumor cells. So is the tumor cells the source of the growth factors that are like attracting the neurons, or is it some other cell in the stromal compartment? We don't know for sure at that point. But when you look at individual cell lines that are isolated from different animal models of pancreatic cancer, as well as from different human patients, you can see that there is transcript for many different growth factors. And you can, as well as in some cases, their receptors. But one thing that you can also see is that there's variability even within the genetic KPC model as well compared to other models or within different patients, which of course could account for some of the variability that we see in the neural changes. But based on this fact that there is this upregulation of growth factor and these hypotheses from clinical data that neurotrophins are important for disease progression, we wanted to determine if targeting this axis and suppressing neural activity through the neurotrophin axis was sufficient to impact disease. So at the time, there was clinical trials going on with anti-NGF for pain. And so we thought it was a good place to start because of the translation ability and capabilities. So we treated the mice biweekly with anti-NGF antibody, and we're able to demonstrate that we suppress neural signaling in both sensory and sympathetic neurons. So just for example, there was reduced expression of the neuropeptides that are released by sensory neurons and a decrease in the receptors that are activated by a nerve growth factor. And in the sympathetics, we also see a decrease in the production of tyrosine hydroxylase, suggesting that our treatment paradigm was sufficient to reduce expression of key signaling pathways in these different nerve types. So how did it affect the actual disease? Well, when we first looked at this perineural invasion, because it occurs early in the disease process, in our control animals, just like in patients, there's a 100% incidence of perineural invasion. But we saw a significant reduction in our anti-NGF treated animals. And when you actually, this graph is just referring to yes, no, there is some cells there. But here we see when we quantify the number of cells that there is, even when those anti-NGF treated animals that had some perineural invasion, it was significantly lesser degree than the control animals. And when we looked histologically at the pancreas itself, we found that indeed in the anti-NGF animals, there's a proportion that actually show no histopathology at all, and their pancreas look completely normal. Which you would never see in the control animals at this stage. And furthermore, when you look for gross metastases, like more than one cell, you can see that there was no metastases at all in the anti-NGF treated animals, but consistent with previous cohorts, 30% of the control animals show gross metastases, and in some cases, more than one metastasis. So it's clear that nerves are associated with disease progression, and that suppressing them, suppressing general non-selectively the neural activity is sufficient to affect these later stage time points. But going back to like my, favorite sensory motor system, the cancer is also associated with sensory motor deficits. And your own colleague, Dr. Rohini Kunar, has done a beautiful study characterizing many different aspects of sensory motor function in animals with pancreatic tumors. And this is just a couple of examples to emphasize that these animals do develop allodynia, which basically means that they are experiencing pain. And they have a reduced activity, just moving around, But importantly, what's consistent with the clinical presentation is that patients typically hunch over and they don't like to stretch out because their abdomen hurts. And so we see significant reduction in climbing, and in other studies, jumping and rearing. And so clearly at this late stage, when there is a clear tumor present, we see these sensory motor deficits. But given the fact that we've seen the perineural invasion and the sprouting occurring earlier during the precancerous stage, I was interested to know what's happening to the sensory neurons at that early time point. And what you can see at that early time point is injury to these animals, to these neurons. And so this is just one example where you have an animal that there was no obvious tumor, but when we opened it up, this is what a normal ganglia looks like. And the DRG on the other side was clearly diseased. And that's confirmed with the histopathology here, where we've almost lost all of the neurons. So when we look at overall the whole ganglia that express or contain the cell bodies of the sensory neurons, at this stage where there's no frank tumor, we are already seeing expression of injury markers such as ATF3. Not surprisingly, it goes up even more once you have a tumor present, but it is suggesting that the sensory neuron changes are preceding the tumor formation. And so because those changes are occurring earlier, are the sensory neurons directly contributing to the PDAC development? And so my goal was to figure out a way to target the sensory neurons specifically. So obviously the anti-NGF didn't do that. And if I went in and I just cut the nerve, I would be cutting, the other fiber types in there. So we don't have a perfect solution yet, but we thought that ablating through a chemical method that targets something only expressed on the neurons would be a good possibility. And so it turns out that the sensory neurons that are in the spinal ganglia predominantly express TRPV1, the ion channel that's important for pain, inflammation, neurogenic inflammation, and it's regulated by growth tract factors. And in the sensory neurons that are in the nodose ganglia, there is still some expression, although not as extensive. So the idea was, could we use this TRPV1 channel that's restricted to these sensory neurons to target sensory neurons and look at the effects in cancer? So we used capsaicin, which is a selective agonist for the TRPV1 channel, and we give a high dose neonatally, and we kill most of the small diameter sensory neurons. And so you can see that we see a significant loss of the neurons when we denervate these animals. And as expected, there's a shift away from the small diameter to a much larger proportion of large diameter neurons, suggesting that we're hitting the population that we think we're hitting. So how did this affect the development of these precancerous lesions or these neoplasias? So normally at that 10-week to 12-week time point, we see these neoplasias throughout the pancreas. And in our denervated animals, the pancreas looked pretty normal. And we were able to see a significant reduction in the low-grade lesions, and there just wasn't enough here to power to detect a difference. But basically, this suggests that it is in fact like an early effect, and that the sensory neurons are doing something to affect this early lesion development. So when we looked at a survival curve and allowed the animals to grow up, we showed that there was a significant increase in survival of these animals, whereas normally they're gone by 6 months. But we did see some cell death and some tumor development. So we decided to take a closer look, and we found that the animals that died early and that had tumors actually had technical failure of the denervation. And so it suggests that that may be the reason why they developed tumors. And the animals that did not have tumors had a technically successful ablation. So this is a neonatal capsaicin, so it's not really helpful for treating patients. And as we saw earlier in the prostate study, there were differences in early versus late denervation. So we performed a study where we gave the capsaicin to the adult animals, and we also found that there was no difference in the incidence of the neoplasias or the tumors, suggesting that it is, again, important early in disease, and later in disease, there's something else taking over. And to our great satisfaction, our findings and observations have been independently replicated. Dr. Pashrika at Johns Hopkins gave neonatal resnifrotoxin, which is actually just a more potent TRPV1 agonist, and showed the same thing, that there was a significant reduction in the incidence of tumors. But he went a step further, and he actually measured that inflammatory area around the tumor cells and showed that that was significantly reduced.